I don't mean to curtail some of the discussion. You know, clearly we had a great start to the afternoon, and just listening on some of the great and animated conversation that we had there over coffee. So moving into this afternoon segment, we've got a piece now really on high-performance R&D. So I'm Jo Pisani. I lead PwC Strategy Ann's Pharma and Life Sciences consulting practice, where I focus a lot on new commercial models and new business models when it comes to pharma. So our topic for today is high-performance R&D. Now, we as PwC do a forward-looking thought leadership series. So it was in 2007 that we first wrote Pharma 2020. And I don't know if many people read that, but it was all around declining R&D productivity, how do we make it more efficient, how do we actually address some of the unmet needs that we're seeing, and how do we actually satisfy the needs of the payer. I think against all of those challenges, we've seen the industry just make tremendous inroads. I mean, if you look at the explosion of science that we've seen within the industry, and Sir John gave us a little bit of a glimpse of some of those things coming through, it's been fantastic. You know, we've got evidence of novel funding models and new evidence of collaboration. I guess the challenge we've got is if we're going to aspire to high-performance R&D, are we actually doing it at the right velocity and at the right pace within the assets that we've got? So are we really incentivizing R&D in the right sorts of areas? And are we really genuinely embracing these collaborative models that we in this conference have spoken about for the last several years in terms of better collaboration? So we'll be looking at how to deliver better medicines for a fraction of the time and cost currently associated, looking at novel technologies and things like biomarkers. And I think particularly acute in the context of what's happened in the UK in 2016, we were very lucky as PwC, so we supported Mike Thompson from the ABPI and Steve Bates from the BIA on the EU life sciences transition strategy, so really coming up with the negotiation points that the industry would want, but also really how to reinvigorate UK life sciences, and also helped Sir John Bell on the Accelerated Access Review. So we were delighted to see the Accelerated Access Review published, 3 weeks ago, and some of the principles outlined within that. So anyway, so without further ado, I'll introduce the panel, and I'll get them to really share their views as quickly as we can then. So we've got Duncan McHale. Duncan's from UCB Pharma, where he's Vice President of Global Exploratory Development, and is responsible for the early testing of novel therapies in humans in collaboration with the New Medicines Therapeutic Area colleagues. Next to him is Chaz Bountra, who I know very well. Chaz is Professor of Translational Medicine at the Nuffield Department of Clinical Medicine, an Associate Member of the Department of Pharmacology at the University of Oxford. He also is Chief Scientific Officer for the Structural Genomics Consortium, and previously had a role in Big Pharma within GSK. So I think we're getting a lot of different dimensions from Chaz today. Next to him is Andy Jones, who regulars at this conference will know well. Andy's a senior leader in AstraZeneca who's moved regularly in his career between project management and a variety of functional line roles, giving him a wide perspective on the issues of working in Big Pharma. And then finally, David Hughes, who's Chief Technology Officer from CN Bioinnovations. Dr. Hughes joined CN Bioinnovations in 2010 as Head of Engineering, was promoted to Technical Director in 2013, with responsibility for the management of all in-house and collaborative research and development. So we'll just start off with some snapshots from each of the panel members on their perspectives on the topic of high-performance R&D. I'll then fire some questions to them, and then it's over to you. And I think we're really looking forward to, quite an interactive discussion with lots of participation from the audience. So please use the app to tweet, and we'll get indications when those questions are coming through, or go for the old-fashioned way at the end when I invite for questions. But I'll open it up pretty quickly. So Duncan, why don't you kick us off with your views?
Thanks, Mr. Okay. I think as you've heard today, and you're well aware, the sort of high attrition rate, the sort of volume clinical development and volume commercialization as a model is failing and is not sustainable. And I just wanted to sort of bring out a couple of things that we're doing at UCB to see how we can address that. So I'm going to start a little bit with the two most fundamental decisions you make in drug discovery and development is, one, what is the target? And then two, what is the patient population? So if we look at the target, where we're going with UCB is, as many companies have, we're sort of saying, well, how can we use the experiments nature has already performed for us to provide us with great confidence that pharmacology will actually provide some benefit? So we're looking at genetics, as many companies are, to identify those targets where you've got higher confidence of having a pharmacological effect that mimics the sort of phenotype you've seen in the genetic condition. But unlike many companies at this point, we're not going into genetics of complex traits and common conditions. We keep an eye on that. But if you actually look at that literature sort of rigorously, and people sort of say the lower attrition rates you get with genetically validated targets or genetically associated targets is actually pretty much all driven by monogenic rare diseases. And in the cancer space, cancers are pretty much monogenic or oligogenic tumors with sort of minimal complex trait genetics into it. So rather than go for complex trait genetics, we've gone to look for rare disease genetics, and we're looking for really rare phenotypes, really rare traits, which are likely to be genetic and which will give fundamental insights into biology of particular systems. So a clear example for us is the genetics of sclerosteosis, a very rare condition, most common in South Africa from a founder mutation. And in this condition, affected children actually quite often present with challenges of skull constraint, challenges with visual problems, challenges with hearing problems, and a lot of bony overgrowth. And then if you look at their siblings, what you tend to get is a group of people and their parents who don't fracture. They may play rugby, they may have high-risk sports, but they never get fractures. And these are families who've got increased bone density. And if you have a child who has a sort of recessive condition, that causes problems. But the heterozygotes, that actually doesn't cause problems, and you're sort of, as I said, protected from fracture. So UCB, before I was there, or Celtech as it was at the time, helped deconvolute that biology and work out that sclerosteosis, or Van Buchen's disease there, was due to a mutation in a condition called, gene called sclerostin. And so what that meant was that if you targeted sclerostin, which is a soluble protein, if you targeted that protein and reduced it, you could increase bone density. So that was used as a key into a treatment for osteoporosis. So that has gone on now and is actually in phase 3 at the minute. But there it's a clear example where you can use rare traits, rare genetics, to actually provide you with a target with high confidence of what your drug will do. And then you can find a condition where that mimicking that genetic condition would be a good thing. And here, postmenopausal osteoporosis was such a condition. So we now have a program where we look at that across a whole range of different phenotypes. And of course, the advent of next generation sequencing means that, you know, compared to when I was a boy and you needed several families to be able to do this, lots of affected members, and we did our linkage analysis first, and then we chromosome walked, and then we got a bit better when we chromosome ran. Now you just get the guys to sequence it, and you can do this off 1 or 2 individuals. So rare genetics for us is a very key piece to actually validating our targets before we take them forwards. And the majority of our targets now have some level of Mendelian trait validation, either from that particular target or a close member of that particular pathway. So that's one area, validation of your targets. The next one is actually how do you choose the right indication? And one of the challenges in the past is we have very much kind of gone for what we thought was the right indication, and we've run with that as quickly as we could, go into 2A, you know, and if we see something that looks like a bit of a hint of a signal, into 2B and generally fail. So what we have done is we've created a system where, in fact, we will always run multiple early studies, multiple POC-light studies, biomarker rich, and we will look at those different patient populations and try to identify that patient population very early in the process. We do accept it can slow down that little bit, and that does feel a bit frustrating at times, but it means that you can go into 2B and 3 with confidence. And in fact, then what we try to do is run phase 2B3 fusion type designs to try and catch up a bit of the space. But that's the area that, you know, we're also focusing on, is how can you find the right patient populations? And like a lot of other folks, we're doing a lot of adaptive designs. We're implementing Bayesian analyses in all of our early development programs, so we can pull as much of that data and information together to try and get the right patient population to marry with what we hope is a drug which we're pretty confident will have a pharmacological effect because of the genetics that we have identified that target with. So that's all a sort of a very sort of key for us. But also we're trying now to think about the data and information we have and we can access in our databases and in publicly available databases to feed back to identify those patient populations going forwards. So we've got machine learning approaches that we can run in-house on our tightly available clinical trial data. And we're now also running that across electronic healthcare records. And there the idea is that you can identify those patient populations which have higher unmet need. Is the predictivity perfect? Not at all, and not what you're expecting. But you can find those patient populations with a lower chance of responding to current standards of care. And that's the group that you want to test early in your development programs, because if you can do something for that group of patients, that's where you want to be. It's better for the patients. It will give you a faster route through to getting launched and to actually get to those patients. Hopefully your phase 2B and 3 programs will be smaller because the benefit risk will be greater. And you will use that as your justification as to why you don't need pre-approval as big a safety database as we might currently be looking at, where quite often we're looking at 3, 4, 5,000 patients worth of safety data before we even launch a product. But if you have a drug which has a really strong benefit, then actually, as a society, we have to learn to live with a little bit more risk and capture that true value data in the real world. And we think this is a sort of a very plausible strategy. It's a very sustainable strategy if we can make that work.
That's great, Duncan. It's great to hear some successes there, particularly around that widening the hopper and sort of the fail-fast type mentality. I'll pick you up on that question later about how we live with the risk, by the way.
Okay.
Okay. Chaz, do you want to give us your view on this?
Well, thank you very much, ladies and gentlemen. Good afternoon. And Joe, thank you for organising this. I'll try and give you a sense of what we do, but before I do that, I'll share with you why we're doing it, because some of you may think we're mad. So I think Joe summed up in a paragraph that what patients want, what carers want, and what society wants is they want better drugs, they want cheaper drugs, and they want them faster. Now, the problem we've all got, and we're all talking about it, the way we're currently doing drug discovery, frankly, it's too costly, it's too risky, and it's too slow. Now, of course, the buzzword always is innovation. You know, we want innovation. Everybody wants innovation. Academia does, industry does, patients want novel medicines. Now, when we talk about innovation, we're saying we want novel targets. We need novel assays. We need novel biomarkers. We need to explore novel modalities. We need novel ways of stratifying patients. We need novel processes. We need novel pricing strategies. And of course, novel means more risk, etc. So that's a bit of a background. So let me just share with you what we've been doing to try and respond to this. So basically 4 things. First of all, what we've done is we have pooled resources. So we currently have had a lot of funding from the Wellcome Trust. So the Wellcome Trust, in fact, have put close to £60 million into my lab in Oxford. £60 million. We get funding from 8 large pharmaceutical companies. Each of them have given us 5.4 million euros. We're now working with 5 different patient groups. We've got funding from government agencies, and we've also got funding from philanthropy. And so what we're doing is we're pooling these resources to share risk. The second thing we do is that we only work on completely novel targets. I'm not interested in working on targets that people have been working on for 20, 30 years. We want to open up new biology, new pathways, new targets for drug discovery. And in fact, many of the targets we work on are targets that people think are intractable, they're undruggable, they're impossible. We work on those deliberately because I believe our job in academia is to drive innovation. And what we do for those novel targets is we generate high quality, novel reagents. So we purify the human protein. I'm not interested in mice and rats, it's always human. We generate assays for that protein. We work out the structure of that protein. We generate small molecule inhibitors for that protein. And we also generate antibodies for that protein. Now, these reagents are incredibly high quality, not because we're clever. They're high quality because we're working with these 8 large pharmaceutical companies. We're accessing their expertise, their infrastructure, their resources, like medicinal chemistry, like developing assays, etc., etc. So we work on novel proteins, generate novel reagents to drive innovation. The third thing we do, and this is probably what makes us most unique, is that all of these reagents, which essentially are starting points for drug discovery, we make those freely available. we give them away to anybody in academia, anybody in biotech, and anybody in pharma, because I believe that's the best thing I can do to catalyze science and catalyze drug discovery. Now, you can imagine the consequence of that is that every academic who comes into my office wants to collaborate with us because they know I've got no secrets. I'll share all of our know-how, all of our expertise, and all of our reagents. That transparency is great for collaboration, it's great for science, and it's great for drug discovery. So we're releasing these reagents to the world, and we're using global academia to crowdsource science and de-risk those targets. The fourth thing we do is that all of our reagents, all of our data, all of our knowledge, we share with the world immediately. Even before we've started writing any manuscript, we share it with the world because we're trying to reduce duplication and wastage. So 4 things: pool resources, share risk, work on novel, impossible areas to drive innovation, make everything freely available to crowdsource science, and release everything immediately to reduce duplication and wastage. So that's all I was going to say.
It's excellent, Chaz. And again, some fantastic, real-life examples there, some really innovative approaches. Andy, you've always got quite a broad view from the big pharma perspective about embracing innovation.
Yeah, hi everyone. For those of you who don't know me, I'm Andy Jones. I lead a group in AstraZeneca called Pharmaceutical Innovation. We sit in the pharmaceutical science area, so I come from a different perspective and spent a lot of my time in the drug development part of the life cycle rather than the discovery part of the life cycle. The group I lead now has some interesting accountabilities. We're accountable for identifying the areas of science and technology where we think we can invest to speed up drug development and create better products. We're accountable for our innovation, insight, and intellectual property processes. And then we have an interesting responsibility in that we act as an incubator for areas of innovation that haven't found a home, a natural home in the business yet. And I have 2 groups. I've had 2 groups this year that we think are really interesting. One is a simulation, prediction, and modeling group where we've used in silico experimentation to try and predict properties rather than drive our knowledge through wet experimentation. And then the other group, which is we like to call the incubator the greenhouse. This is growing out of the greenhouse now. It's a digital health group. So the goal of the digital health group was to see how we could create digital tools that would allow patients to optimize the use of our medicines to maximize their benefits. As we've got into this, what we've realized is that there's an opportunity to use digital tools to improve the way that we do R&D as well. And one of my sort of mantras as I've gone through development is there's no money in an NDA. One of the problems that we have in pharma development, I think, is that it's too compartmentalized. We have a big milestone when a molecule moves into development, and we have a big milestone when a submission is approved. But for me, the reality is when you can, you really succeed, is when you can get a molecule to a patient, and when you can really start to impact a patient health and do that across a big population. So one of the things that we're looking at now is how we can use digital tools, not only to improve the speed that we do development, but actually how we can improve the outcomes of our drugs by gathering data in the clinical trial phase. One of the things that I think is really interesting is if you look at pharma R&D, as opposed to R&D in any other industry, much of pharma R&D is targeted at getting that approval. We are doing trials to demonstrate safety and efficacy to get approval. If you look at the world and the way that other industries do R&D, they do R&D in order to develop a product that a customer, in our case a patient, really wants. Yeah? And they provide that service to the customer of something that they really want. One of the things that we're really interested in is what data can we gather from our clinical trial experiments in a digital form that we can then use to better support patients. So just to give you an example, if we can track side effects digitally in a clinical trial and we could identify a cohort of patients for oncology therapy, for example, who might suffer from diarrhea or rash, some of the common side effects of oncology therapies, if we can identify what type of patient can develop those side effects, we can then look at supporting that patient when that patient becomes a commercial user of the product. So one of the things that we've become really interested in because of our background in digital is how we can look at different data sets, how we can combine genomic data sets with data sets that we can gather from patients. So we talked about asthma patients with high levels of eosinophils. Can we combine that with data that we collect on how patients use their inhalers, or what environmental factors trigger exacerbations, to help us treat asthma patients better, to understand which asthma patients should be treated with expensive biologics versus generic inhalers. So one of the things that I think is going to be incredibly powerful going forward and will drive a step change in R&D productivity is how we combine the data sets that are available to us as proprietary data sets with other data sets. And I'm known for being pathologically optimistic. I actually don't believe that the healthcare companies will the tech companies will step into the healthcare space. I believe that the real value here will be in deep collaboration between tech companies and healthcare companies, where we bring our experience and they bring their experience, that we can data mine, understand the data sets that exist, and predict better outcomes for patients.
Yeah, okay, that's great. Thanks for that, Andy. David, over to you.
Thank you. So my name is David Hughes. I work for CM Bioinnovations, and we're a small company. We were spun out of Oxford University in 2008, and we're a maker of what they call human organ on a chip products. So essentially advanced in vitro tests for drug discovery and preclinical work. And what I wanted to talk about today was how I think that improved preclinical tools can assist in improving the performance of R&D overall. And I'm going to exemplify that with our own field of human organ on a chip, and then move on to some slightly broader perspective on this. We like testing things on human cells because we believe that's a highly relevant thing to do. But you know, we're in a hard field, and the pharmaceutical industry has and continues to do a large amount of animal testing. And I wanted to say a little bit about this. Now, first of all, not to say that testing on animals is a tool not to be done, and it has led to some excellent innovations. But to give some stats around this, the NIH, 47% of funding by the NIH features some component of animal testing. And this is a market that makers of novel tools like us would like to break into, but I think that stat just illustrates how well embedded existing processes are in the pharmaceutical industry, and perhaps how difficult it is to get a new tool into this kind of market. So just to say a little bit about the kind of technologies we work with. So what we do is we take human cells and we put them in an environment in which we hope that they accurately recreate the physiology and pathology of human health and human disease. And this is an area that's had increased funding in recent years. So there's been funding from the European Union, the NIH, and DARPA, and indeed we're part of a DARPA-funded consortium which aims to build a human body on a chip. A rather ambitious and perhaps slightly misleading thing to say, but we're working on that and we're getting there. But even in spite of high levels of funding from governmental organisations and considerable interest, I would say, from pharmaceutical companies, we found getting uptake of these tools into pharma R&D still remains a challenge. And I think, to echo something which all of the rest of the speakers have said, one of the reasons we find this is a challenge is that I don't think yet we've found really great ways of engaging in collaborations between small spin-out companies such as us and big pharmaceutical companies. And I think that that is an area that really working on more closely will assist us in getting these what we hope to be novel tools into the hands of the right people, and ultimately helping to develop some better medicines. So whilst I'm passionate about human organs on a chip, I don't think that they're going to solve all problems. I'm not naive in that. And really, I think the opportunity for new tools is really, in fact, in the synergy of many of the exciting new preclinical and discovery tools that are coming out of universities and pharmaceutical company research labs, and in bringing those kinds of tools together to enable a holistic approach to the problem. So we have things like CRISPR for gene editing, we have stem cell derived lines to create models, we have the vast amounts of clinical and genomic data which are being brought together in, for example, the genome projects we heard about earlier. We've got omics type work which is opening up a wide array of information that wasn't available previously, and we also have the ability to bring these data sets together through modelling. And I think really for tools in this area to make a considerable impact, there has to be an integration of all of these parts together, because I think it's only through integrating these parts together we will be able to achieve a deeper understanding of the biology, which will enable us to pick better targets and ultimately to have a more productive R&D process.
Okay, that's great. Thanks a lot, David. So I hope you agree with me. I think we've seen some really interesting ways in terms of challenging the traditional R&D model, right from the science to the open innovation, more collaborative parts, to actually looking at innovation in the service delivery being enabled by things like organs on chips. I guess I was going to mention this question on risk, because I guess all of these and any innovative step with it carries a degree of risk. Be it risk in terms of taking a lot of products through into into phase 2A, be it risk in terms of giving away your IP and where the money might lie. Andy's risk, I guess, would be, you haven't got the IP of a molecule to protect you there. It's almost the proposition of a service around that. So really interested in the panel's view about how we can manage risk, or ideally embrace risk and use it to our advantage. So Duncan, did you want to kick off?
So yeah, I mean, the aspect I'm going to pick up is risk in terms of benefit risk assessment for patients, risk in terms of patients. So we convince ourselves that by having data packages of 3,000, 4,000, 5,000, 6,000, larger and larger data packages, we are getting greater and greater confidence that the drugs that we put out there are safe. Now, in truth, the incremental confidence in safety is a lot less than the perceptual confidence. So if I give a novel agent to the first 50 to 100 subjects, I get a reasonable feeling that I'm not going to do anything acute, we're not going to do anything particularly severe in a sort of a very acute type of setting. And in monotherapy, as we start to get bigger and bigger, we get a little bit more confidence. We got to 12 months, we'll get some confidence. But once I've got to 800, 900 subjects to 12 months, to really make a big difference in terms of being confident on the safety, you need to be in the tens of thousands, if not more. And so one of the things is that we need to probably, as a society, think about how are we prepared to take a little bit more risk in terms of putting molecules into the clinical setting, into the commercial place, in the clinical setting, with smaller data packages, but on the flip side of that, use electronic healthcare records, use data infrastructures to pull the safety data back and really learn about it. And that's important for a couple of things, because actually randomized clinical trials are good for efficacy, they're good for common safety events, they're poor for effectiveness, so does it really work out there in the real world, and they're poor for longer term, rarer safety events, because that's not what we do. And one of the classic examples is if warfarin is one of the commonest causes of adverse events presenting to hospitals. You want to be really disappointed? Run a head-to-head study with warfarin thinking you're going to beat it on safety. In a randomized clinical trial, it's hard. It's hard because the people who go into clinical trials are very motivated, they get their testing, they get their dose right. You need to put it out there in the real world. So I think safety data in the real world is much, much more useful, and something that we need to aspire to reduce the development costs and start getting actually meaningful safety data. Now, this is an uncomfortable place. It's an uncomfortable place from our regulatory colleagues who have for many, many years been that arbiter of benefit risk to say this is safe enough. And we're asking them to say this is safe enough with a smaller data package, accept and we'll collect more data. It's an uncomfortable place for companies. So don't get the idea that pharma would like to push out these molecules with a lot smaller sort of data sets, because the worst thing at the minute that you can do is launch a drug and pull it back. So again, from a pharma perspective, it's an uncomfortable situation. But from a patient perspective, from a sustainable biomedical research, new assets perspective, I think it's where we need to go. As a society, we need to get more comfortable with that little element of risk there and sharing our data more to truly understand the benefit risk in the real world, because that's what we care about. Most people are treated in the real world. The benefit risk assessments from a phase 3 randomized controlled trial are useful, but ultimately they're not what you need.
Yeah, okay, that's great, thanks. Chaz, did you want to comment?
I mean, I have to say, I agree with Duncan. I mean, I believe as a biomedical community, we are great at certain things, but we're crap at others. I think we're not good at target discovery. You know, there's 7,000 rare diseases, there's hundreds of chronic diseases. Each of these chronic diseases is probably 10 different diseases. And there's 22,000 proteins that could be a target for drug discovery. Our ability to say that this novel protein, if I modulate it, it's going to work in this disease or in this subset of disease is appalling. This is why our failure rate in phase 2A is 70, 80, 90% for novel targets, etc. So I believe the industry has become extremely good at certain things. It's become very good at doing lead optimization, toxicology, regulatory, the really big clinical studies, the phase 2B, phase 3. But target discovery and phase 2A are our biggest problems. So what we've tried to do to de-risk target discovery is 3 things. 1, I didn't mention this earlier, we are picking high priority human genes that have come out of large-scale human genetic studies, and we are generating our reagents for those because we believe those are going to drive translational studies both in academia and in industry. So that's one way of de-risking. The second way of de-risking is we're generating very high-quality inhibitors and antibodies for these novel proteins and then crowdsourcing science to de-risk those proteins. The third thing we're now doing, and one of the advantages of sitting in academia surrounded by hospitals and clinicians and patients, is that I can much more readily access patients and patient material and patient databases than if I was sitting inside GSK or UCB or whatever. So we're now accessing primary human cells from healthy individuals and patients, and we're now testing our molecules on those because I think that is a much better way to come up with new targets for drug discovery. So those are 3 things we're doing to de-risk. I should say I absolutely believe that target validation only happens in patients. It doesn't happen in animal models, it certainly doesn't happen in cell lines, and it probably doesn't even happen in primary human cells as well. So let me just share with you one anecdote. So 8.5 years ago, when I went back to Oxford, I was approached by some venture capitalists in Australia. They asked me to help them develop a pain asset that had come out of the University of Melbourne. And at that time, all they had was some animal model data. And as some of you know, I dislike animal models. I think largely they're a waste of time. I don't think they predict what happens in patients. We spent 4 years convincing ourselves that that molecule would work in patients. We then started a clinical study 3 years ago. 18 months ago, the data came in. This was a phase 2A study. We dosed this to post-herpetic neuralgia patients. We had 183 patients, 91 placebo, 92 active. We dosed the compound for 4 weeks. And at the end of the 4 weeks, however we cut the data, the compound has worked. And last year, Novartis bought the compound and the company for 700 million plus, etc. But now let me tell you the target. So you're all aware angiotensin can work at 2 receptors, AT1 and AT2. If you block AT1, you get antihypertensives like losartan. This compound is a blocker of AT2. It has no activity at AT1. The day we got that clinical data, I went home and I told my wife that it had worked. And I said to her, had I stayed in GSK, I would never, ever have persuaded GSK to do that clinical study. Because everybody thinks, oh, angiotensin, it's cardiovascular blood pressure. Nobody thinks of neuroscience or pain. This is where terminology is blinkering our thinking, and we have to be completely data-driven.
Okay, great. Talking about data-driven and service provision, Andy, do you have a view on how you manage that risk?
I actually wanted to talk about something else in terms of risk, and it was something that you talked about, you mentioned earlier, which is the risk around IP. I think if we truly are, going to get high performance R&D, what we've all recognised, I think, is we're going to have to get better at collaborating. And I really applaud what Chaz says around being as open as possible, being as transparent as possible, driving collaborations and the exchange of ideas. But one of the biggest hurdles that I see is constant frustration with collaborations being frustrated by discussions around intellectual property. And I think fundamentally there is a huge number of misconceptions about the value of intellectual property to someone and the risk involved in sharing ideas. We spend an enormous amount of money funding academic research. We fund the most basic academic research. We fund case students for PhDs. I've been in the organisation nearly 30 years now. We've been funding case students for nearly 30 years. I've been deeply involved in it all that time. I can honestly say in all that time, I don't think one idea has come out where there's been genuine intellectual property. There's been fantastic science. There's been huge amount of collaboration. We've identified some really brilliant PhD scientists. Is there any real IP that has come out of that? Probably not. And yet, my group spends more of its time trying to get IP negotiated with universities around case students than anything else. It's a small world. Yeah? The other part of it is that people think that big pharma is out to steal everybody else's IP. In the world is small, if a pharma got a reputation for being unfair with small companies' IP, the collaborations flow would stop. Everybody respects the ideas that people generate, and everybody recognizes that collaboration is an absolute key to kickstarting R&D. But we have a huge number of people who are obsessed by IP. I was head of chemistry, chemical development for AstraZeneca. I said to our IP lawyers, we're no longer going to fund process patents. I'm not going to pay to fund any of our process patents any longer. You can shut that department down now. They said, we won't have any intellectual property protection of our processes. I said, if chemistry hasn't moved on in 20 years, that's the least of our worries. But people have unrealistic views of what intellectual property is worth. Yeah. And we have to get beyond that, get scientists talking about the real value of research, and move into how can we collaborate better. And IP is important, and when we get to a point where we know there's something worth protecting, we should sit down together and understand what's a fair deal for everybody involved. But if we waste 6 months at the start of every collaboration discussing some hypothetical IP that might be generated, we're never going to have a high-performing R&D organisation.
Yeah. Okay, that's great. And Andy, I know you mentioned you're an internal optimist, but I think you're getting a lot of nodding heads around the room on some of those statements. If I can turn to the audience now, and I think we have been getting some questions coming in, Vicky, haven't we? I can hear them in my ear. No, I can't. I can see them up here. Right, Graham. Oh, hang on. I can see you're very happy, Graham. There you are. Why do pharma still waste millions on flawed market research? Sorry, we're down at the bottom. It'll scroll up to drive what? Okay. Right. Flawed market research to drive R&D when newer proven methods exist. So do you believe the industry is using flawed market research to steer R&D programs? Duncan, you're desperate.
Well, I agree. I mean, not being in the commercial sector, I agree. It's all flawed. I mean, I think to be fair, I think that market research was a tool people were comfortable with, and they felt it provided some value that was regionally specific and would help because you can sort of target which particular regions you're interested in and do the research there. I think probably what you're alluding to is we're moving into a digital world. Actually, you can pick that market research up in many different ways. If it's harder endpoint type work, can you do it through electronic health records? Can you do it through claims databases? Is there ways you can do it that way? I think for softer research, can you do it through Twitter? Can you do it through Facebook? Can you do it through, digital media? And I think most of the companies are now experimenting in that area. It is an area that makes our compliance officers a little uncomfortable because it really is sort of a little bit wild out there, and it's difficult to know, what the compliance laws really are. Are you really talking directly to patients, or in which case are you falling foul of certain regulations, etc.? So I think that we're in a learning phase in digital media to learn both what is the compliant, ethical way to reach out and get information from patients using these approaches, and what was a robust way of doing this as well, because as you all know, not everything on the internet is absolutely true, and so you can easily sort of pull back things and create hypotheses which actually probably aren't necessarily true. So I think we're also trying to build rigor into getting patient insights through things like digital media. So I agree, market research is limited. It's not necessarily flawed, you just need to understand the biases it's giving you. I think digital media, digital approaches have huge opportunity, and we are learning how to do this, but it's not straightforward when you think about compliance, when you think about rigor and application. So I agree that's the way we're going, and we need to go there faster.
Just a comment, John. Sorry, Justin.
Well, no, you go first.
Okay, good. So, I mean, again, this is a very personal view, but one of the things is I think for a long time we've been confused about who the customer is. If you're doing market research, you want to understand who the customer is. So, Joe and I were talking about this earlier in the week. When I started, late 80s, we essentially sold products to doctors. That was the deal. So, the doctor was the customer. Then in the early 90s, when Pharma went through a period of difficulty getting molecules approved, we got a kind of edict around regulatory focused development. So the FDA became the customer. And then Pharma went through a period of getting things approved but not being reimbursed. And then it was like, oh, well, now it's all about the payer. And 2 or 3 years ago, I got quite depressed because everybody was talking about patient centricity. And my first thought was, oh no, this is the next big thing. We've gone from doctors, regulators, payers, and now it's patients, and that'll disappear. But then I had another kind of optimistic moment and actually said, no, this is absolutely right. It is patient centricity, and people talk about patient centricity all the time. You have to think about the words. Patient centricity. So that means that the patient is at the center of something. What are they at the center of? They're actually at the center of a healthcare ecosystem that involves healthcare providers, payers, supporters, and pharma. And so they sit within an ecosystem. So one of the things I think in the past we've been confused about the customer is, and if you're developing a product, you don't just have to think of what it means for one person in that ecosystem. You have to think how this product will sit in the whole healthcare ecosystem and how it will be used. And I think once we get into that mode, we'll be a lot better at designing the products that will actually get used.
Yeah.
So, I mean, I grew up in GSK, and this is a company that's made a fortune in the respiratory area. They've sold beta agonists and steroids for decades, etc. And I remember in the early part of this century when they were launching a combination product, basically a long-acting beta agonist with a long-acting steroid, Seretide, those commercial forecasts were wrong. So even though they'd been in the area for 2-3 decades, they knew all the clinicians, they knew what was wrong with the existing drugs because their drugs were the gold standard. We still got the forecast wrong. Now the future is we're going to develop products for newer diseases, rare diseases, subsets of disease. We're going to be paying for outcomes and not products. We're going to have products where we charge different amounts in different countries, etc. I think the future is just getting more complex, and frankly, my confidence in commercial estimates will be even lower.
Right, no, I think that's absolutely fascinating in terms of that view. And I guess, when we look at it from an investor perspective, we did a piece of work earlier this year where we saw a lot of venture capital money was almost going into the sort of the safer areas, I guess, where there was almost a herd instinct of saying, yes, we can understand this. So all the money was going into immuno-oncology and also in CNS, but really it was into Alzheimer's and Parkinson's, still highly neglected areas such as depression that weren't really covered from CNS. And things like musculoskeletal were actually very poorly invested. So Duncan, it's great to hear what you're doing in that area as well. Maybe I'll open it to the audience now, just to try and keep people on their toes and steer the mic. So we've got a lady in the middle there, and then we'll go to this gentleman over here. So perhaps the lady in the middle first, and then we'll come to you, sir.
Good afternoon, Sylvia Lobo. A philosophical question for you. Do you see a difference between high-performance R&D and optimal performance R&D? And if so, what does that look like, and how do we rebase?
Yeah, I'll have a go. Philosophy is not really my thing, but I'll give it a go. I think that where you get the discussion between high-performance R&D and optimal R&D is when you come into the discussion around how much money do you have and where do you want to spend it. And that's generally the factor that's used to optimize R&D and optimize risk investment and the extent to which you front-load or back-load a development as well. So I think, one of the things about pharma R&D is it's incredibly difficult, and sometimes I wonder, wow, it's amazing we actually get anything to the market. So I think you can create an incredibly high-performing R&D organization. I worked when Zeneca merged with Astra, I worked on a team that said, how could we get from identification of a molecule to launch routinely in 5 years? Not submission, but launch. It actually wasn't that difficult if you had a bottomless pit of money. So I think the thing where the interesting discussions around high-performing versus optimal come around the discussions of when you invest in order to accelerate. So we were talking about the supposed demise of pharma and the low performance of R&D. We just launched a lung cancer treatment, Tagrisso. We got it from phase 1 to launch. I can't remember the numbers now, but it's less than 4 years. It's the fastest development I think ever, but I wouldn't claim that, to be honest. But we absolutely knew it was a winner from the first trial. Actually, I was involved in the development of Crestor. We knew from the first trial it had a better profile than atorvastatin, and that was an incredibly easy drug to develop because there was no debate about whether you invested. We knew there was a $5 billion market. We knew that we had a differentiated product, so you could really perform in those cases. But it's optimal R&D is around understanding the risk and when to invest. And for me, that's when you get into discussion about high-performing R&D versus optimal R&D.
Yeah.
But Andy, just to be provocative, I think 10 years ago, I would have said they were, maybe 15 years ago, they were potentially different. I did work for Pfizer here, for my Pfizer colleagues, so I'm not taking the name in vain. I would have said Pfizer in the 90s and early 2000s was an optimal R&D organization. It took molecules which were flawed but were precedented mechanisms. It made great drugs. It sold them. I don't know that I would have said it was necessarily high-performing research side of that, because I think a lot of those were precedented. The chemistry was great, biology less so, but it was optimal because it took those pieces, fixed them, and drove them to patients very effectively, very efficiently. Merck, you could all argue, was more on a sort of science, less on the commercial side or that side of it. I think now you need to be both a high-performing research and development organization, so the science has to be great in terms of the molecules you make, the biology you understand, the patient populations you go into, and you've got to be optimal in terms of you've got to be effective to get there quickly, because it's a very crowded world out there. So I actually think right now my answer to the question was no, I think you've got to be both. But I think there was a world when you could be optimal without necessarily having to be at the highest end of all of the science and pieces.
So I'm not convinced I agree with Duncan. I mean, as an industry, let's face it, we've not had a new drug for Alzheimer's since 2002. We've not had a new antibiotic since 1984. And some people argue we've not had a new drug for psychiatric disease for the past 3 or 4 decades. Our failure rate in phase 2A for completely novel targets is anywhere between 70 and 90%. You know, nobody would invest their own money in this industry. And this is not a high-performing industry. So I'm not sure how I can answer the question whether we want to be optimal or high. I would just like us to be better. If we can just reduce that attrition in phase 2A from, let's say, 80% down to 70 or 60, that will have a phenomenal impact.
Yeah, okay. I'm conscious we've got loads of hands, so maybe we'll motor quickly through the other questions. So it's this gentleman over here.
Mike Capaldi from Synergos. I had a question about risk and regulators and culture. Now, Duncan, you made the point about people need to be prepared to take more risk, but of course one of the problems is Joe Public litigates when things don't go the way that they expected. And in terms of risk, do you think it's the regulators that are risk averse, or actually do you think it's the companies that think the regulators are going to be risk averse and therefore continue to do lots of testing that maybe they don't need to do if we had a less risk averse approach?
I think it's both. I mean, I think there is, and whether it's a mutual conservatism or whether it's kind of each, the sort of industry thing. Well, the regulators are going to be very conservative, therefore we'll be equally conservative and get bigger and bigger packages. But undoubtedly, both the regulators and the companies would become uncomfortable if you started to have significantly smaller packages from a safety perspective. But I don't think it's one or the other. I think it's a ecosystem, it's a culture that's there, that is also to a degree in the medical profession as well. If you actually talk to individual patients, and it's always difficult because you haven't had the adverse event, but if you talk to a patient and said it was a 1 in 1,000 chance of having, a significant liver injury with this drug, the majority of them with severe conditions would take the risk. Now the problem is if you're the one patient, you're the one, and therefore how do you also get society to accept that you live with the risk as well, and you can't just have a very litigious kind of society, so that, as you said, we get everybody comfortable with it, but then as soon as something happens, you sort of can sue them as well. So it is, for me, neither the regulators or the industry. I think both are conservative, and I think there's a culture of conservatism that we need to bring forward. I actually think if you talk to patients, there is an appetite for that, but it does need to be, kind of led with actually a sort of informed conversation, because it's very easy to say I would take the risk if you haven't actually, had the event.
Yeah, it's probably a question for Guido in the next session as well. There's a question down here, so this gentleman in the front row here.
Could I also respond to the last one, Joe? Would you mind? So, I mean, I actually think somebody talked about culture in the previous session. I do think biotechs in the US are less risk averse. Secondly, I think when we make decisions by committee, we always end up with mediocrity. You will never end up with the extremes, the really risky stuff. I absolutely agree that society, I think, are unreasonably expecting squeaky clean drugs. And we as scientists know there's no such thing. And if I took a teaspoonful of salt every day, that would have consequences. And we know that when we take drugs, we're basically managing the intake of a poison. So I was very disappointed when Vioxx had to be pulled from the market. I think Merck got themselves into a corner, but I think that drug clearly has no GI risk, but it had a cardiovascular risk. But the cardiovascular risk is the same as all the non-steroidals that are still on the market today.
Yeah, no, that's right. Okay, let's go in front row.
Hi, I'm Sampreet Ramachandra from Fresenius Kabi. And I want to build on some of the points that you faintly touched, Joe and Chas. And I want to direct this question to somebody who is outside the industry, better still, someone who has stepped out of the industry. when you meet R&D professionals, it's very easy to hear about things like oncology or unmet needs in orphan drugs. You very rarely hear about things like pain, anesthesia, analgesia, where there hasn't been a new drug in the last 3-4 decades. There have only been small modifications. So when we talk about R&D performance, not just better, cheaper, or quicker, how do we align R&D efforts with, in terms of with the societal or patient needs, and away from just pure economic returns that the industry expects? Or for such innovation, would we have to just rely on white knights such as yourself sitting outside the industry?
So I think one of the things that really worries me is that in biomedicine, there is massive duplication. There are so many academics, so many biotechs, so many farmers. They're all working on the same few ideas. They do this in parallel. They spend 6-7 years coming up with a proprietary molecule, and when they go into the clinic for the first time with a novel target, we know the failure rate is anywhere between 7 out of 10 or 9 out of 10. Most of our early ideas fail. It is a waste of money, it's a waste of people's careers, and actually it's a waste of patience. And you could argue it's ethically and morally wrong. So that's one comment around duplication. I think your comment around patience is incredibly important. Patients in the future are going to be great influencers on governments as to where they invest their research dollars. And they will also be heavily involved in terms of which drugs to pay for. You know, I think one of the lessons, just a few days ago, I was saying to Joe just in the break, I had dinner with David Atschuler last week. So David is the head of R&D at Vertex. So Vertex have largely spent most of their R&D money on cystic fibrosis. And they got a lot of money from the Cystic Fibrosis Foundation, a great partnership. But you know, that gene, that transporter, we learnt about that in 1989. And Vertex launched the first drug in 2012. 23 years it took us to get a drug to the market for that one condition. We talked a lot about genetics. Huntington's disease, we know the protein, we've known it for more than 2 decades, where's the drug? You know, ApoE in Alzheimer's, we've known about that since the early 90s, where's the drug? The sodium channel blocker for pain, NaV1.8, the genetic data is really strong, where's the drug? I think we need to make this process a lot faster.
Okay, thanks for that. Now, the red light's flashing at me at the moment, so I'll sum up just now, and I will ask each of the panel members just to give their final word of advice to each of the companies in the room here, just as a quick one sentence. But I'll attempt to sum up. And I mean, I was very much buoyed up about this conversation. I think Andy's optimism must be infectious. As Mike pointed out earlier, you know, key to the UK really surviving through Brexit and thriving is to make sure we really make the most of the UK assets that we have in life sciences. And I think we've seen great evidence today about how that approaches. So we've seen, you know, innovation in terms of science, particularly with adaptive design and the role of digital pulling it through. Chaz talked about very innovative innovation models around pooling resources, giving away reagents and sharing IP immediately within that. Andy talked about innovative service design and putting the patient at the centre. And then clearly David's got the innovative clinical tools to help drive that, and also really another plea for effective collaboration. I also heard very strongly the plea to be better about this as well, and I think that's probably something that we can all embrace and drive on. So I think in terms of, you know, as a panel session, I hope you found it as interesting as I have. I'll each of the panel members now one word of advice about how to get to high performance R&D, or at least optimizing R&D, to the lady's question here. So Duncan, a soundbite, please.
Okay, you've hit a little bit of maybe doom and gloom wasn't meant to be. I think we are near of huge optimism and potential. The biology we understand is progressing so rapidly, so fast, versus, you know, where we've been in the past. When I started this industry, it was small molecules and kind of proteins were just about there. If you look at your potential therapeutic options now, small molecules, antibodies, cells, you know, peptides, nucleotides, the tools we have to be therapeutics is just sort of infinitely better than it was 15, 20 years ago. And the information technologies around being able to tie together information and create knowledge out of it is just unbelievable in terms of how fast that's going. So I think we are in an area of incredible potential. But I think the winners will be the ones who can collaborate the best, share bravely, and actually move forwards with therapies which will really make a big difference to patients, because nobody is going to be good enough in all of these areas, but there is huge potential out there. So I think collaborate bravely.
Great. Collaborate bravely. Chaz.
So just to follow on from that, what I would like to see happen a lot more of in the UK, and I think it happens in the US, is that people work in academia and then they'll go and work in Novartis and then they'll go and set up a biotech and then they'll go back to MIT. And I think that's the sort of stuff that we want. And I think if we did that, we would understand each other's perspectives better. I think collaboration would be easier. I would advocate more open collaborations. It allows us to leverage a lot more resource and share risk and make the whole process faster. I think there are certain things that I would do less of. Less animal models, be less risk averse, and less decision making by committee.
Great. Thanks for that. Andy.
Yeah, three things really. The first one, I would say, focus on the needs of the patient, but recognize that the product will be used within an ecosystem. The second one is to be absolutely passionate and unremitting around collaboration. And then the third one is to be optimistic, because I think we've heard, although this is a difficult industry and we face challenging times, we have made some truly fantastic advances over the last 20 or 30 years. And sometimes, you don't see the grass grow. I go home every day, the grass doesn't seem to be grown, but by Sunday it definitely needs mowing again. We're making progress. It's a long job, but I look back over my career, I've been blessed to have worked on 3 compounds that have made it to the market and made enormous difference to patients' lives. So let's be optimistic. We work in a great industry, and what other industry can you think of where you do the coolest science and impact on patient lives? Yeah. It's just the best industry in the world.
There we go. Great call to action. And David, your soundbite, please.
I'd like to second Chaz's call that we perhaps become a bit more American in the way we do this. In that interchange between big pharma, biotech, academia, I think that's very important for exchanging ideas. I'd like companies to think about working in real, true partnerships, particularly with small companies where both sides are bringing the skills that they have. And finally, I'd like to continue to see strong funding for science and R&D post the Brexit.
Yeah, that's great. Well, thank you. I hope you found that invigorating conversation. I mean, collaboration was a huge theme, and what better conference to have those collaborative discussions over the next coming days. So please join me in thanking the panel for their insights today.